Yeah. So it's vaginal delivery, so the natural birth of a child. So if you're born by vaginal delivery instead of cesarean section, then we show that they have a 14-fold higher potential of getting colonized with these bacteria. So that's quite a strong, interference factor. Then exclusive breastfeeding is also important. So if the child is getting formula during the first 2 months of life, then we see a very much reduced colonization with these bacteria. And then the third thing is that having older siblings also tends to modify or increase the colonization with these bacteria, also during the first 2 months of life. Actually, I think it's quite important that you have that you don't see an inhibition of the IgG, because the IgG is there to protect us against the different infections that we are acquiring during, also during early life, but all through our life. So if we see an increase of that or a decrease of that as well, that might have had some implications in terms of infection burden and so on. So having a selective inhibition of the IgE, which is, so IgE is connected to allergic diseases, but also, of course, to the ability of handling a parasite infection. But in this case, we didn't see a total IgE inhibition, but an allergen-specific IgE inhibition, at least in the children that we followed longitudinally. So I think it's important that it's selective. Yeah, so that means that you can either prevent by supporting the growth of these bifidos if they are there. So you can support them by adding in substrates that they live off preferentially. So that is human milk oligosaccharides that you get in breast milk. Or you can supply specific, you could call them probiotic supplements to those kids that doesn't have enough colonization in the very first weeks of life. But another strategy could be to include aromatic lactates into specific supplements, so that you don't increase the bifidobacteria, but instead you give the specific metabolite. So there are different means of now intervening if one can see that we actually can draw causality based on the newer study that we are now setting up. What we see is that it seems to be very important for the early colonization with these aromatic lactate producing bifidos that you deliver naturally, so by vaginal birth. But you can, of course, also introduce these bacteria to a cesarean section born infant by giving a slurry of the fecal matter from the mother. It has been done in a few American studies, or studies conducted in the US, where actually the fecal slurry was provided into breast milk and then provided to the kids. So this is also a way of introducing it to those who might not have it already. And then the other thing is one should maybe be careful not to give formula supplementation to the kids at least before 2 months of age, as we can see that this is, really also having a great effect in terms of reducing the colonization with these bacteria. Yeah, so one thing is that it is important that it is a prospective study. So we don't, we cannot claim causality. We have only observations that tend to say something about associations or a link between the early colonization to later disease development. So one needs, of course, to make intervention studies to understand the mechanisms behind this and whether there is a causal link. So this is actually to convene on these intervention studies to make sure that supplementation to, for instance, C-section born infants would provide them with the right colonizing bifidobacteria in the early life, right? We did replicate some of the major findings in other cohorts, but at the moment it's only cohorts from industrialized countries. So it would be interesting to see how it holds true if one is looking into other communities around the globe. So at the moment it's replicated in a German cohort, and then the original cohort is from Sweden.